All right, let's get started. Can someone confirm you can hear me on your end from the mic? All right, thank you, Rachel. All right, hello everyone. My name is David Ruck. I'm here at the University of Maryland in Baltimore, Maryland, and this is Cytometry in R. So, Cytometry in R is our little weekly mini course aimed at those who have some prior flow cytometry experience, but who have no to little coding experience before this class. However, it doesn't matter where you are as far as what your previous flow cytometry experience is or what your previous coding experience is. Everyone is welcome to take this course. So, some of you may be wondering is why do we put effort into even learning how to do cytometry in R? And there's a couple reasons for this. So first, if you think what's been happening with our field in the last few years, spectral is rapidly growing in terms of the total number of fluorophores we're able to handle on a day-to-day basis. So we're past the 50 barrier, we're heading on 60, 70 markers that we can have within a spectral flow cytometry panel. And this is kind of similar to the situation we already have with mass cytometry panels. However, unlike the mass cytometry panels, we can acquire lots and lots of events with our spectral flow cytometers, so we end up with massive data sets that are both very, very large in terms of number of markers, but also in total number of cells. So it becomes a lot more complicated when we're trying to figure out what is changing within the data for these experiments that we're acquiring. And we are no longer able to do so solely by manual gating alone. So we do have to start relying on some of those unsupervised analytical techniques to try to figure out how this works. And the particular reason why those unsupervised analytical techniques normally get introduced to our field as R packages is partially due to a founder effect. So the Bioconductor project started in the early 2000s. The people who wrote the initial bioinformatic tools to analyze flow cytometry data were part of the Bioconductor project, so everything has grown and has been a contribution of R package and R packages. So now, even if you go to Cyto, every year you'll see there's a couple Python packages. I saw a Julia and a Rust package last year. So there is a little bit of growth in field as far as other programming languages, but the vast majority of R's supervised analysis continues to be in R. Now, if you happen to be at one of the institutions where people are already using R for their flow cytometry analysis, you are set. But if you're at one of the institutions like mine, where our bioinformaticians have no idea what to do with a flow cytometry file, you're kind of out of luck. And what makes it more difficult is there are very few resources online to learn how to do it. You will eventually find them if you're searching for a while, but when you do find them and you do encounter them, a lot of them tend to be aimed at those who have intermediate R experience or aimed specifically at an established bioinformatician who's just trying to learn how to do the flow cytometry side of the analysis. So if you are working at a flow core or you're just starting off in a PhD program, you're left kind of like thrown off the deep end and you're not knowing how to swim and how to work things out. So this was the exact same situation I was when I started learning R back in 2020. So I had just finished my qualifying exam, and the next week afterwards, COVID shut down the campus and we were all sent home. At this point, I only had 2 experiments to my name, so it wasn't like there was a data set I can analyze within FlowJo at that point in time. But we had an RNA-seq data set from the lab that we had gotten processed by some company. The results didn't really match with what we knew the biology was behaving in our own system, so they handed the data set to me and they're like, here, figure out how to learn R, reanalyze this RNA-seq data set, and we'll move from there. The next 3 weeks were like the most miserable in my life. I tried downloading a script somewhere I found on the internet. I tried running it. I got packages installed, but it would continually error out, give me warning messages, and I just could not understand what was going on. Eventually, 3 weeks later, in just frustration, I just called my PI and asked, please leave me a data set on a flash drive outside my door. There's just no way I'm going to ever be able to work in R. Now, in the benefit of hindsight, what I realize is even though that script would have ran perfectly fine and given me the dataset analysis that I was looking for, and even though I had installed all those packages perfectly fine, the original script didn't do one important thing, which is call library for one of the packages that's used further down in the script, which is how a R package gets attached into a local environment in your computer, and your computer knows what's there and is able to use it. Now, if I had been at an institution where somebody already was doing coding in R, and I could have reached out and gotten help, this would have been a relatively small barrier to entry, and I could have continued. However, because I was navigating entirely on my own at this point, I was very much a fish out of water, and there was not much I could do. So this is one of the reasons we are doing this course. First off, it is a passion project, but I have learned how to do cytometry in R the hard way by literal cycles of trial and error, eventually figuring out how to do things. So I do have a good context now as far as what's happening within the language. It's no longer so much of a black box. So I'm like, okay, we do need a good course of how to do flow cytometry in R that covers a lot more topics, especially for the community with everything growing as it is. But we also really, really need a class that's aimed at those who have no flow cytometry experience and that teaches how to do R at the same time as it's reaching flow cytometry. So that will be part of our goals in this course. Now, some things the course will be and some things the courses will not be is I don't want to just give you code that's cookie cutter where you just plug and play and get results out, because that's kind of the same situation I was trying to be in back in 2020. What I want to do is more for this course is teach you how to build the tools that will help you get past any barrier that either R or flow cytometry throws at you. So if we're thinking of this in the context of like you're back in the Middle Ages, you're trying to take a castle, I could just hand you a ladder to get you over that wall. Or I can teach you how to build your own ladders so that if there's a taller wall, you can build a slightly taller ladder. Or I can teach you how to dig under the wall, or I can teach you how to make high explosives to blow the wall to smithereens. So that's kind of the mindset I'm bringing for this course. And just like any other thing you're trying to learn, regardless if it's trying to learn a new language, if it's trying to learn an instrument, if it's trying to learn how to draw, it takes time and it takes practice to get good at things. So if we think of our daily practice as flow cytometrists, mass cytometrists, we don't just cobble together a panel, see if it works on the first try, and if the unmixing is horrid, we don't just give up and say, we're terrible flow cytometrists, this is not going to work, I'm going to find a different field of practice. Same thing, if you're doing your first experiment and you mess up your pipetting, it doesn't mean you'll never be able to pipette, it just means you need to have more practice at pipetting. So when we're thinking about can we code and can we not code, I don't want us to think of things in such a binary. I want us to think about it as we're going to be going through cycles of we try something, it doesn't quite work out, we eventually figure out how to get it to work, and then we move on from there. And then slowly but surely we'll be able to do things that we weren't able to do a week, a month, a couple months ago. So that will be the aim of the course. Now, when I first envisioned this course, I was only thinking I would have maybe 10 or 20 people online interested. So we have had a lot more people sign up, which we are ecstatic for. We're glad that we identified an area that everybody's excited to learn about. But we're going to have to change a few things as far as the online format. So I won't be able to, like, say, run this line of code, wait for you guys to run the line of code in your web browser, then come back to the view for the YouTube, and then go for the next line. So what I'm going to try to do, especially for these 1 hour of time, I'll be conscientious of what I need to do. I want to give you guys the big picture. I want to provide the code. I want to provide the explanations behind the code, because that's the most important thing that's missing of trying to learn on your own journey. And then slowly but surely, we'll build out each topic each week, putting the building blocks together until you're able to build your own pipelines for flow cytometry analysis entirely on your own. These notes are for housekeeping. We are running our course off of GitHub repositories, and the useful thing about being in a GitHub repository is I can update the data sets throughout the week, and then when I upload the new data that's available for next week, if you have forked, which is basically carbon copied the course repository to your own GitHub account, and then brought that forked copy of the repository to your local computer, it's only like 2 clicks of a button to bring in all the new data sets, all the new code, any bugs fixes I made for last week's lessons without having to track things down individually. So that's one of the reasons I want to use GitHub for the course. The other one is we're using version control, which we'll talk about more next week. but then also we're using the discussion page on our GitHub repository kind of as a community forum. So if you go and check it right now, you'll see a lot of people who were taking up one of the other live streams this week have already run into some installation issues, and we're troubleshooting those there. Same thing, if you want to talk about things more in depth, the community discussions page is kind of like building some of those in-person elements that we're not able to do virtually. So please head there, create a GitHub account if you haven't done so already. We have a bunch of pre-course materials already available on the website, and now the initial live streams are online. You don't have to do them all. You just really need to have your GitHub account set up and get through the workstation setup for your own particular computer operating system, regardless if it's Windows, if it's Mac, if it's Linux. The other extra pre-course walkthroughs are more just giving you a broad overview of what the class infrastructure is, and we'll be covering those bits and pieces throughout the course. So those are not an immediate worry about this week's scenario. I would focus on the GitHub account, your workstation getting set up, and then installing the first few packages that you'll need throughout the course. And with that, let us begin. So R is a programming language that has been around for a while. It started back up in the 1990s, and it started being a little bit more used in the early 2000s. However, a lot of the growth that we're seeing within the use in R has happened in the last 15 or 20 years with the introduction of the tidyverse packages. And the tidyverse packages have rounded some of the rougher edges that used to exist within the base R language, which is why it's able to be used a lot more readily by beginners. That was the same repeat of the message. And part of the reason why R has grown so much is because you have the ability to make additional R packages. So you can have a cool idea of something that you want to do. You can write the code basically to do a particular thing. These are what we know as functions within R. And then get all your functions together and make your own R package. And this can be added on top of the existing R software to do cool and interesting things that you wouldn't have been capable of doing before. Now, there are thousands of R packages available now thanks to the many efforts of all the different individual developers over the years. And as a result, the R ecosystem has grown very, very widely as far as what's being able to be done. So it doesn't matter if you're working right now in flow cytometry, or you're an ecologist, or you're working in political science. A lot of the analysis that's happening and a lot of the visualizations you're seeing in publications do have R involved, at least in some part of the process. And one of the things I like to point out is not all our packages have to be very serious, very heavy lifting, doing a particular computationally heavy task. Our packages can also be made for fun. So a couple examples I like to point out are, there's somebody who made a R package that's basically the little snake game, and it'll have it pop up every time you're waiting for your R code to run on the recalculation screen. And there's a big collection of R packages that are literally just dedicated to setting color palettes and themes based on movies. So if you wanted to turn in your next report and have it with Wes Anderson movie themes, you can do so. If you want it to be Studio Ghibli themed, you can do so. Or if you're a young parent and you have kids, you can literally make them individual reports with bluey colors by particular characters. So there's a wide variety of R packages there. If you think there might be something there, it's likely there is already. Now, all our packages start off as source code, which is what we'll be learning how to write in this course. Now, source code is very much human readable. However, your computer is not able to interact with it directly. So what happens between the source code and you downloading it and installing it is your computer compiles it into what's known as a binary format with which your computer is then able to interact directly. As everything is being brought together within a single package, if you have created a completely new R package that only relies on its own code to make its tools, i.e. the functions, it's able to work self-contained. It doesn't need or rely on anything else. However, the useful thing about R packages is you can borrow other people's code, other people's functions, and have them do particular things within your own R packages. In doing so, you're able to do things that you were not able to do before, and you save yourself the hassle of having to rewrite all their code. But at the same time, you introduce a dependency on having their R package installed. So when we're talking about dependencies later today, this is the concept of the R package you're trying to install is borrowing something from another package. Therefore, that other package needs to also be installed before the package you're interested in can successfully build on your computer and be used. Now, there are over 23,000 R packages spread out all around the internet. So trying to track down where on the internet each of these individually is would be too much of a pain to deal with. So R packages are collected together in what are known as repositories. And these repositories serve as the intermediate distributor. So you don't have to reach out to the individual developer's place they've stored it on the web. You just reach out, your computer will reach out to, like, let's say the main repository, CRAN, and say, I'm looking for this package. CRAN will then check to see if it has the package. If it does, it will return it to your computer, and then the installation process can proceed. So within context of R packages, there are three main locations we go when we're trying to download R packages. There's the main repository CRAN, which has over 23,000 packages. There's the Bioconductor project, which has about over 2,000 packages. These tend to be more focused on the biomedical sciences. So when we're downloading cytometry packages, often we're going to Bioconductor. And then there are individual R packages that are hosted by their developers on GitHub. These tend to be the ones that are newer and that they're in process of getting submitted to either CRAN or Bioconductor. But if you're looking at a R package on a poster or in a new publication, oftentimes these will still be available via GitHub. Now, the thing that was very tricky trying to figure out on my own back in 2020 and 2021 was how you go about installing an R package. The commands that you use to do so will vary depending on which of these repositories that R package is actually located at, which makes it confusing because there's three sets of commands for each of the three repositories, and you need to make sure you're mix and matching correctly. And then at the same time, you also need to know where's this R package stored in the first place. So the main focus of today is like walking you through this, taking back the curtain to be able to like understand a little bit more of how you go about doing this, how you go about troubleshooting this process. And we'll also be touching on the concept, which is where I was having issues back in 2020 because I had not attached the install package to my local environment, which is done via the library call. So between both of these points, we'll be talking about most of today. And let's get started. So there's a little bit of background keeping. You can read these notes within the website itself. The main thing I want to emphasize for this course is if you're opening in new material that you have brought into your local Cytometry and R fork on your local computer, I want you to copy and paste any data that you're actually using into a project of your own making. So if you're opening a positron for the first time, create a new folder, and then within that folder, just move and copy and paste anything that you need. This will prevent any conflicts next week when you're bringing in week 2 data if you accidentally overwrite something within like the actual course project itself. So we'll revisit this throughout the course. This is just a general head up if you start tinkering on your own. Now, the other part to Positron is it's multilingual. It can handle both Python, R, and a bunch of other languages. So at the very start, you need to make sure that you select R right here on the interpreter in the upper right side of your screen. If R is correctly installed on your system, you'll see this little pop-up down here in the console, basically telling you what R version is installed and providing like help file, license locations, different things of this sort. And just as general overview, I go more in depth as far as the different components of this in the Positron pre-course walkthrough. But the big overall picture of the different parts of what you're seeing in the software, the console window, which is where you actually write the code and it goes and runs it and returns the outputs, is down here in the panel section. When you're editing files, this will be popping up here in the editor with each file appearing as a tab. There's the primary sidebar, which is dictated what you're seeing based on which of these little tabs in the activity bar is selected. And then on the right side, you have the secondary sidebar, which is where different objects, different variables that are created when you run code appear as lists. And then if you generate any figures, those figures will also appear as plots in this section here. And we'll be going through this throughout. I'll be mentioning these locations. This is just to familiarize you guys with the terminology as far as where are things located within the positron window. So as I mentioned, once we install a package, we need to make sure it's loaded into the local environment when we open positron or any of our IDEs that we may be using. That makes it active and allows the computer to see what functions, i.e. what tools are present, so it can then use them in turn. One thing I want to emphasize is how to figure out information about a particular function. Now, when I say a function, I mean a particular set of code that does a particular thing think of it kind of like a tool in the toolbox if the toolbox is your R package. So the big giveaway for what a function is they'll always have these little they'll always have these little parentheses at the end. So you'll have the name of the function first appearing here, then followed by the little parentheses, and this together basically says, hey, run this particular tool, and within the parentheses, you provide any arguments that you need to have executed. So in this case, we'll look at the session info function, which is kind of how we just see what packages have been attached to the local environment since we last opened our Positron browser. And for this course, as you're copying and pasting back from the website, if you hover over a code block chunk, what you'll end up seeing is something that looks kind of like this, where there'll be a little copy assigned that will let you work with it. Give me a second. My computer's acting up here. There we go. There'll be a little section where you can like copy and paste it back, and this helps you like prevent having to retype very long sections of code or accidentally mistyping something. So it's just a little bit makes your life a little bit easier across the board. All right. We have a question on the chat about RStudio. Will it be sufficiently different from Positron if they need to switch over to Positron for this course? So Positron and RStudio are made by the same company, Posit. Positron is one of the newer versions. It's come out within the last 2 years. The reason we're using Positron is it's a lot easier to use in a lot of aspects than what RStudio is. So a lot of the things that used to be hidden like several menus back are right where you need to see them. They're quite obvious. Setting up with GitHub is much easier. Finding those help documentation files are very much easier to do. And there's a bunch of things that YAR also allows you to use multiple languages if you wanted to. So because they're made by the same company, all the things that those who work at Posit have seen over the years as being trouble points have been fixed and patched in the newer version for Positron. Also, I'm concerned that RStudio is not going to be maintained long term. That will be discontinued starting after next year, at which point I would have to rewrite all the documentation. And at least anecdotally, when I was teaching people how to code 2 years ago using RStudio, it took a lot more time to teach them how to use it than I've been teaching people how to code with Positron in the last year. It's a lot more intuitive, it's a lot easier to pick up, so that's part of the reason why I am trying to use Positron as much as possible in the course. Similar situation happens if you are somebody who does a little bit more coding already and have used Visual Studio Code. Visual Studio Code Positron is a forked version of Visual Studio Code with a bunch of the RStudio components added on top. You can probably mimic what you see for Positron if you're already somebody who extensively customizes what you use, but it's a little bit harder to do than just installing off the bat. So I hope this provides a little bit of context as far as the decision as far as why we're using Positron. Obviously, if you're a company and you're not able to install Positron, we'll figure out what workaround for you guys in that case. But if you're able to get it installed, I really recommend it because what used to take several months of trying to get familiar with the software only really takes a week for people I've been teaching in the last year. So if you're looking up information, trying to find a help file for a particular function, there's a couple ways that you can do it. If you are working within Positron, you can just hover over the function, and the function help file, if it's present, will pop up, and then you'll be able to read. Now, help documentation in R is only as good as the person who wrote the function. So if it's going to be, you have a good collection of help files that are completely nebulous and leave you more confused than when you started, but there's also an equal number of help files for different packages that are actually good as far as explaining how to use things. So I'll point you how to find these internal resources, but I'll also show you how to find some external resources on the websites that will be useful in the long term. The more traditional way, especially if you're not in Positron and you're trying to look up the help information for a particular function, is you can do the little question mark sign and then put the function name and the parentheses, and this will also pull up the help file. And when you do so, generally, it'll pop up here on your right sidebar, and then you'll see the name of the package, the name of the function, all the different details about the different arguments that are present within the function itself. And then if you scroll to the very bottom, you'll also see this little index tab, And this basically goes to a dictionary of function names for that particular package. So you'll see in this case for the utils package, it starts listing alphabetically all the different functions that can be found. Now, the vast majority of these you'll never really need to interact with daily, but if you do decide you need to find something and you can't remember where the function's name is, but you do know it's in a particular package, going via this index route can help narrow down the possibilities as far as which functions you're looking for at that stage. So the very first function we'll be working with in this course is the session info function, which is basically how we're going to be able to see what's attached to our local environment, which is what our computer actually sees and what our computer is actually able to use as far as the R packages that we may have on our system. So if you run session info, you'll get an output within your console window that looks something like this. It'll vary a little bit depending on if you're on Windows, if you're on Macs, or if you're on one of the Linux distributions. But the important thing for this course when we're looking at it is I want you to scroll down towards the bottom. So in this case, I have just opened Positron. I have just run the session info function. I have not attached any library. I haven't called library and attached any R packages yet at this point. So you'll see the only thing that's currently showing and listed is this attached base packages, and it lists 7 different packages. When you download R and you're downloading the R software, you're actually downloading these 7 packages that come by default and are always active anytime you have R open. So you may occasionally see them as the stats, the utils, and the base packages as far as dimensions go. And then because I'm working in Positron, Positron also has a few packages it keeps ready in reserve in case you're trying to do something. In this case, you'll see these are not attached, meaning your computer is not able to use them directly. However, they are in a category that's called loaded as a namespace. So your computer is aware that these packages exist, but it doesn't know what the contents of the packages are as far as the functions or the tools that are present inside. Versus in the case of an attached package, it knows that they exist and it knows the entire list of functions that are present within each of these packages. So if you write a line of code that says, find me this particular function from this particular package, your computer knows right off the bat where to go, where to find it. It doesn't have to go to an intermediate step. So let's see what this looks like as we go throughout the day and we install packages and then we load them via the library command. We'll see how the sections that we just saw using the session info package change as more things get attached to our local environments. So we'll be starting off by installing packages from CRAN. Now, CRAN is the biggest repository of them all. It's over 20,000 R packages. And in the context of the Cytometry and R course, we'll be going to this particular location primarily when we need an R package that does general data manipulation, like changing things from rows to columns, working with matrices, data frames, that kind of nature. Or if we need them to do particular visualizations as far as the plotting aspect. So if we were talking about a tidyverse package, we'll be going to CRAN when we're trying to install them. In this case, we'll be installing first the dplyr package. And the dplyr package is located on CRAN, so we'll need to use the CRAN-specific installation command, install that package's parentheses, when we are actually trying to install dplyr on our computer for the first time. Now, you can look up the same help information that we saw for session info by either hovering over it in Positron or adding a little question mark in front of the function name. If you do, the help file will pop up. The main thing I want to emphasize when we're trying to do the installation command is this is the function, install.packages. The arguments that we're providing it are everything that's present within those parentheses. So when we're doing a quotation marks dplyr, we're basically telling our tool, the install.packages function, hey, go to CRAN and search for the name of the package that's present within these quotation marks. If you find it, please download it and bring it into my local computer. Now, one of the things that's always a continual struggle point for anybody getting started in R is inevitably we forget quotation marks. And then at that point, we have an error that looks something like this. The reason for this is R, when it doesn't see quotation marks, thinks, oh, this is an object that's already present that I created in my environment. So it goes and checks your local environment. It doesn't see any object called dplyr, and that's why it returns an error at that point. So if you're trying to install anything from any of the repositories, always remember the install commands need those little quotation marks in order to work properly. So if we remembered our quotation marks and we put them present, when we go for install.packages, this is what's going to happen. You'll see all of a sudden your computer console window come to life. You'll see a bunch of text appear, and then it'll just start rapidly scrolling faster than you're able to read until all of a sudden towards the bottom you'll see this done sign in red with a download source packages are now present in a particular file path location. So I don't want to go through all these different steps as far as what's happening. I'm not a computer scientist. I don't want to be a computer scientist, so I'm not interested in all the little steps. But I want to provide you enough context that if something goes wrong on a package installation, you know how to go about troubleshooting it, and you know kind of like the basics of what's happening in the background. So when you tell install.packages, go download dplyr from the CRAN repository. It first goes and checks CRAN to see, does this file actually exist? When it sees that it exists, it proceeds to download it to your local computer. After it's present on your local computer, it first needs to unzip the file, and then it needs to start doing the actual installation. After it's done installing, it wants to check that, make sure that yes, it's installed correctly, meaning that it can be attached to your local environment and therefore it can be used and all the functions within it can be used. And if that test passes, then you'll see the sign of done and within parentheses the particular package that has been installed. And then that original zip file that got brought in from CRAN gets moved to a temp folder, which your operating system will then delete at some point later on in the future. So if we have already installed a dplyr, we need to then make the next step, which is making sure it's actually attached to our local environment. Everything in R in this case, once you've installed it, when you're trying to get it to actually be present and all the tools available for you to use, this happens via the library function. And as always, you can look it up by either hovering over it in Positron or adding the little question mark in front. When you're looking at library, what you're going to see is you don't need the quotation marks, and this is where the annoyance is, especially for beginners, because install commands need quotation marks, but the library command that's also using the exact same name of the package does not. So when you use a library command, just remember you don't need the quotation marks present. So let's go ahead and call library for dplyr, and let's go and check behind the curtain to see what's happening when we have quote unquote attached it. So you can see all the base R packages are still attached. They're always going to be present as long as R is active in your environment. But you can now see that after we've called library on dplyr, is now appearing under this other attach packages command. So at this point, your computer knows that dplyr is present. It also knows all the different functions that are present within dplyr. So if you use them in a particular piece of code, it's going to be able to use them. Now, dplyr has its own set of dependencies, i.e. other R packages that it needs to function, but those don't necessarily need to be loaded at this point in time. So you can see these are loaded via the namespace, but they're not attached, meaning your computer is aware that they exist, but if it was going to start actually using them, it needs to go through another step of attaching them to an environment first. So this list will continue growing, and you'll see more of a spread as you add more packages each time. So one of the main functions within the dplyr package, i.e. one of the main tools that the dplyr package uses, is the select function, which basically allows you to grab a particular column within a matrix. Now, if you have loaded dplyr and attached it via the library command, if you do the little question mark select, your computer is aware that there's a select function because it has already loaded the dplyr package. So you'll start seeing dplyr appear in the list of suggested options that then pop up. In a situation where you had not loaded the library for dplyr, you wouldn't actually be seeing the select command because your computer doesn't know that those tools are present within the dplyr folder. This is a situation kind of like what you'd see for the ggplot package, which I have installed on my computer, but I have not yet called via library. In this situation, if you did try to look up the ggplot function, it's going to give you back this error saying, hey, I have no idea what you're talking about. I can't find it. And the only way you'd be able to actually look this up would be you need to library parentheses ggplot to be able to attach it to your environment. Additionally, sometimes some packages will have a way to get to kind of like a landing page on the help section. So you can just put like question mark in front of the package name, and if they do have it, it'll get you kind of to this landing page with the author information and useful links and the index. Not all of these have it, so when we're installing PicoQC in the next step for the Bioconductor project, it doesn't have a landing page, so you won't actually see it if you're trying to do a question mark name of package in that case. And we don't really have to unattach something in R for most of the time. Here's the command if you were really curious of how to do it. Basically, anything you can do in R, you can reverse it. But I want to show what happens in the context of we have just loaded dplyr, but then let's say we go ahead and detach it from our environment. You'll see at this point, if we go to the next slide, if I then try to look up dplyr, I'm no longer able to get to that information because it's no longer attached to the environment. So remember, you can install your R packages, but then once you have them installed and you're about to work with them, you need to actually load them, attach them into your environment using the library command so that you're able to use the functions within. And this is what I didn't know back in 2021. And if I had been able to troubleshoot that, I probably would have had an RNA-seq analysis done in a few months at that stage. All right, the other way is there's a short workaround we'll encounter later on in the course where basically you tell your computer, hey, I know this is present, I know this function name is there, go grab it. And this kind of involves using two little colons right next to each other in your particular line of code. I'm just introducing this right here, but we'll come back to it later on in the course when we actually need to use it. And just like this, you can unattach something. If you're installing a package, you can also remove a package. I don't anticipate having to do this very frequently. Oftentimes, you're needing it if you need to have you're having a particular package having a dependency conflict with each other, in which case you can remove it the same way you install it, using the remove that packages function, and remembering your quotation marks, the name of the package that you want to uninstall. Now, as I mentioned, beyond the annoyance of does this command use parentheses or not, the main pain point for somebody getting started is this package from CRAN, or is this package from Bioconductor, or is this package from GitHub? So in this case, we're going to install the next package, which is PicoQC package, which is used for quality control of our FCS files. And it is located on the Bioconductor repository. So I want to show you what happens if you try to install PicoQC using the CRAN-specific commands instead of the Bioconductor specific commands, what kind of error output you're going to get from this process. So if we try to do it this way, what we're going to eventually get is that same kind of can't find this particular version, it's not loading, please find somewhere else. So in this case, if I try to do it, install that packages, oh wait, 1 second, went a little too far on that one. Didn't pop up today. Okay. All right. So if we're trying to install something from the Bioconductor project, from the Bioconductor repository, it's a lot less packages than what we see in CRAN. It's about 2,000 something packages, but most of the flow cytometry packages we'll be using are located within this repository. Now, Bioconductor needs a package installed from CRAN first. That will then allow you to network and interface with the Bioconductor project, and then being able to bring in the particular packages that you need from Bioconductor. So the way this works is we need to first install the Bioc Manager package, which is located on CRAN. So just like any CRAN package, we do the install.packages command, remembering our quotation marks, the name of the package, in this case Bioc Manager. And after it's installed, of course, we need to remember to first load it in with the library command. So we go ahead and library Bioc Manager, this case without the parentheses. And then if we were trying to figure out information about it, we could do so via the little question mark install option. And now at this point, the package specific command used to install things from Bioconductor is not install that packages, but it's install. And install still uses the quotation marks. And this is what's going to allow us to go to the Bioconductor repository and download those Bioconductor specific packages at this page. So when we do this for PicoQC, what we see is it has a few additional dependencies. So you'll see these dependencies being listed right up about here. So you can see it has RJSON, it has clue, it has complex heat map. So the very first step in the installation process looks a little bit different from what we saw for dplyr. Basically, for each of these dependencies, it then goes and searches CRAN or searches Bioconductor, finds those dependency packages, and also downloads them at the same time. The scroll then gets a little quite numerous and rapidly flying by the screen, but basically what's happening is the same kind of installation that we saw for dplyr is now happening for each of those individual dependencies. So in the case of like RJSON gets downloaded, RJSON gets unzipped, it starts the installation process, it's then tested to see if you can attach it to the environment, and then you get the done exclamation sign. And then as you continue for each of those other dependencies, you get the same kind of sequence happening again and again. until ultimately all the dependency packages that PicoQC depends on are present, at which point you can then see PicoQC begins to installs and installs correctly, and you get the done sign, and all those little temp folders that are the zip files get sent away for eventual deletion. Now, occasionally when you're downloading a lot of packages, you'll get this little pop-up that says, hey, some of these packages that you have new versions. Do you want to update these packages? And then it gives you options of update all, update some, or update none. Now, if you're downloading something from GitHub, it will look slightly different, but it looks kind of similar in as far as it'll give you a numbered list corresponding to particular options, and then you need to specify the number that you want to use and hit enter in your console window to be able to move ahead. Part of this, it depends on how much time you actually have. Like if you're rushed on time, it's fine to skip it for the time being. But if you have extra time and want to update, please go ahead and update. As you can see, if you're looking at the package version numbers, most of these changes seem to be relatively small changes, so it's not really going to affect your code on a day-to-day basis as far as if you update or if you don't update. And then once you have downloaded a package and you're ready to proceed, remember in this case we tried to install a PicoQC. We need to load it into our active environment using the library command. So once we library PicoQC, if we check our local environment, we can see we now only have Bioc Manager and PicoQC because we had detached dplyr up above. And then you can see PicoQC's dependencies are also now present within our environment and ready to be used if they're actually needed at that point. The reason not everything is attached versus just loaded via the namespace but not attached is often different packages will have functions with the same name that will do different, completely opposite things. So, for example, dplyr has the select function, but the flow core package, which we'll also be working with later in the course, also has a select function, and they do completely different things. So you need to specify which of the two selects you're actually trying to use in a particular context. So if you loaded all 200 different dependency packages that are present when you're doing your script, there'd be a lot of these kind of conflict issues and you would have unpredictable results, which is not good when you're trying to do things reproducibly and replicably. So this is why you see the situation where it's loaded via namespace, meaning your computer knows it's there, but it's not actively brought into your environment yet at this point. Now, if you're also curious not just what's attached, but what packages you have actually installed in the past, there's a function for that. It's the install.packages command, which will then allow you to go in and actually check to see is it present or is it not there. All right, at this point we have covered, let me double check on my computer. It's glitching a little bit. What's going on here, bud? There we go. Now, at this point, we've downloaded packages from both CRAN, which is the main repository, and Bioconductor, which is the more specialized repository. So the last one we need to cover for today is the GitHub repository. Now, GitHub is not actually a repository in the same sense of CRAN and Bioconductor that just collect all the different packages together. GitHub is very much, our packages are found there, but they're found under individuals, people's GitHub accounts within a GitHub repository folder. So when you're trying to download something from GitHub, you're going to that person's account, and then you're going to that person's repository and downloading that particular location. Additionally, not all packages that you found on GitHub are gems. Some of them are, and they're really great packages that are always been present on GitHub and that are not available on CRAN or Bioconductor. But a lot of packages that you do find on GitHub, you'll find them mentioned in a publication. You'll see that there's a repository, but then you'll see they have not been updated in years. And then when you actually go and download it, you'll find it's not actually working. This is because that the functions that are used and the dependencies that are present within those packages change over time, because our packages are not static. If you find there's a bug, you fix the bug. If somebody finds there's an error in the documentation, they update the documentation. If somebody wants to add a new feature, they add a new feature. So they're always evolving a little bit over time. So if you're trying to install something from GitHub and it's not working, just double check when the last updates were done, because it's possible it's an abandoned R package and you may have to do a lot more troubleshooting to get it to work. So kind of similar to what we had for the Bioconductor project packages, we need to download an intermediate package from CRAN first that will then allow us to go and download packages directly from GitHub. In the case of Bioconductor, this was Bioc Manager. In the case of GitHub packages, what we're working with is the remotes package, which is available on CRAN. So when I'm working through the course material, you'll see these spot check kind of things, which are like if you're reading through the website and you want to test your knowledge of what we have just covered, this kind of gives you the question of like, how do we go about installing a package from CRAN? And I'm kind of using these little drop-down sections of code as the reveal the answer option. So if you're trying to install something from CRAN, you'd use the install that packages command, and then within there, you'd remember use your little quotation marks and put it around the name of the package, this case being remotes. Similarly, once you have installed that package, you need to attach it to your local environment. How would you go about doing so? If you were testing yourself, you could check your answer and then see that yes, you'd use the library command surrounded by parentheses, the name of your package, without a particular set of quotation marks. And then finally, if you're trying to look up a specific information about how to install things from GitHub, which is the install._GitHub function, how would you go about finding out the help information? Well, if you look at the reveal answer via the code dropdown, you'll see that yes, there's the old version of !install_GitHub, or if you're working within Positron, you can just hover over that function once it's been called by library and be able to access the information. So, endpoint, we've installed dplyr from CRAN, we've installed PicoQC from GitHub. What I want to do for our example from Bioconductor, what I want to do for our example from installing from GitHub is I want to download a small R package, which is the flow spectrum package. It is made by Christopher Hall, which some of you may not know. He's responsible for the small series of tutorials on how to do flow cytometry data analysis in R. These YouTube tutorials were very helpful when I was trying to learn back in 2021, and I really like his flow spectrum package because it does these little kind of density spectrum style plots that you would see like on an aurora. So it's a good small self-contained example that we can use for our showing how to do the process of installation today. So the main things to remember when you're trying to install from GitHub is you need the GitHub username of the person who has the package that you're trying to install. So in this case for Christopher, his GitHub user account is Hallie166. Then you would include a /, and then you would include a repository name. So in this case, this would be flow spectrum. So if you're putting together an entire installation command, it would be install_GitHub, parentheses, quotation marks, username / repository name. And then we would run that. Now there's a little bit difference that you'll see in the opening scroll of text when you're downloading something from GitHub versus if you're downloading something from CRAN or from Bioconductor. Namely, you'll see this opening section of things. And part of this is when you're downloading something from GitHub, you're downloading actual source files. Versus when you're downloading from CRAN, it's already been compiled into a binary format that's directly pastable by your computer. So what your computer first needs to do is it needs to compile that source code into a binary format, at which point it can proceed to do the regular installation. This compilation process is what's done if you're on a Windows computer by the RTools software I had you install. If you're on a Mac OS, this is done by that developer tools section that you also had to do an installation for. So if those were not present, this would partially error out as far as the installation process goes. All right, at this point, I've shown you how to install packages from CRAN, from Bioconductor, and from GitHub. So when I was working through this example for the course, I didn't have very many errors that I encountered. So I want to show you how to go about the process of figuring out if you do encounter an error, given how many people we have taking this course, what the process steps of trying to narrow in about what particularly might be going wrong with that particular installation. So if you see an error, oftentimes first thing you do is you'll look through the sprawl of text that pops in the console, and you'll try to see if you can first spot that little error sign. That's the first cue of focus your attention down here. And then in this case, what I did is I installed one of my packages from GitHub, and then I removed one of the dependencies that was needed for it. So when I tried to reinstall it at that point, what's happening at that section is, you can see I tried to install the package called Araba, but the particular dependency I had removed artificially is missing, which is what's causing the error. Now, this boiler warning plate message that you get during an error is not actually the solution to your problem. So in this case, you can see it's suggesting go to install.packages as the install command. But Luciarnica is not on CRAN, it's on GitHub currently. So if you try to just copy and paste this help suggestion, it's not going to quite work. But at least once you've located where in this location it is as far as the error message, you can kind of get a little bit more context cues about how to go about trying to troubleshoot it. One good thing that you can do is just kind of like copy and paste this particular portion of the error message and put it into your search web search and see what comes up. Given that how many people we have installing software, I do ask if when you're installing the list of software we need for today, if you do encounter a library installation error, please take a screenshot and then go to the discussions page and post it on the discussion page. One, I will help you troubleshoot like what's going on with your installation of that particular package. But then two, we can build better examples for updating this particular web page documentation so that anybody who's taking the course after the course concludes and is having these same kind of issues has a few more examples for which they can work with. Now, as I was mentioning, there's the concept of package versions, especially when we're looking at those update. Do I want to update? Do I not want to update? This is a little bit less explained, so I want to make a little mention of it. So if you're looking at the session info when we're trying to see what's attached, if you're looking at the actual packages, they have these numbers. It's like, for example, for PicoQC, it's 1.20.0. This is basically the package version. And the reason we have the package version is because these packages change over time, and we want to be able to, for reproducibility and replicability, be able to go back to a particular version and see if it was working at this particular point in time. So the version numbers kind of help us to note at which point in the package's evolution are we talking about so that we can be able to work with it. So also another way beyond just session info to see this information is the package version function. So we can do quotation marks, PicoQC, and then within the package version function, and it returns that same set of information. Similarly, if you're trying to find a citation for referencing a package in a paper, you use the citation function. So same thing for PicoQC, it pulls up the citation, but then you can also see the particular version down here. How do these version numbers work? Well, there's a couple ways to think about it. So the very first number and the version number is reserved for major changes. Let's say somebody came in, completely rewrote the package, gutted the internals, rewrote the function names, so that basically it's no longer back compatible with the code that was used for the last version. That's considered a major change. So you'll notice for like dplyr and for PicoQC, they're still on version 1 because all the changes that have happened subsequently have been more in the minor add-on kind of style versus a complete rewrite of the package. That second number is the minor version. So this will be like I add a new feature to a particular package to do something new, or I change things a little bit to be able to use spectral flow cytometry data or imaging flow cytometry data. I haven't changed all the functions and tools for all the rest of the package. I have just added new functionality. So that's a case where you'll update in the second number, the minor change. And also Bioconductor is on a twice yearly cycle, so you'll see these updated like twice a year. So in this case for PicoQC, you can see it has grown significantly as far as being on Bioconductor for a longer period of time. And then that last number is often just reserved for like if you have a bug fix and if you need to change things out. So Gabriella has a question. Do we need to install packages every time? No. So you only need to install the package once. But once you've installed the package, you just need to make sure you attach the packages to your local environments. So oftentimes what I'll do, and you'll kind of see this in some of the work-true examples, is there'll be a code block that has the install that packages commands to install everything. And then once I'm done with that installation, I'll deactivate that code block or just get rid of it entirely, just keeping the library code block to load in individual packages. So yeah, you just need to install them once. You don't need to install them every time you load in. That would take a significant amount of time. So we're not going to go too far into the version numbers. I just want you to know that they exist. There's a possibility that in about 5 weeks or so, when we're using the ggcyto package, we're going to have to install some particular dependencies to get it to work. So I just kind of want to introduce how to use these versions to be able to install a particular time point for a particular package in time. So if you're trying to install something from CRAN that's older in a particular version, you'd use the remotes package, and then you'd be using the install_version function. In this case, if you wanted to install, let's say, ggplot, you include the name of the package within the quotation marks. You specify the version number within the quotation marks, and then basically Repos is telling it go to CRAN to download from here. Similarly, GitHub's a little bit more varied because it's not as formal in terms of the actual version numbers. So version numbers often will be when you're looking at GitHub packages actual like releases. So if I was trying to install a particular version of my Lucianago package, I could go back and say, go for this particular release, which I had called v0.99.7. However, if a developer doesn't do releases, you can always go back and check when that Git commit was happening. And those Git commits have specific hashtag numbers, and you can use that in place of a version number to say, I want to download this package as it was the first day of Cyto last year to see how it has changed over time. All right, at this point, we've covered two of the main pain points, namely the use of the quotation marks, and then attaching things to the library, and then figuring out where things are located at and the install specific commands for everything. So we have looked at a couple examples of the help files, namely by doing either the question mark in front of the function name or just hovering over it in positron, but not all the data sets, not all the help file that we have for a particular package is just what's contained internally. Many R packages have external websites that do a lot better job as far as explaining how to actually use their package. So I kind of wanted to walk you through the process of how do you go about finding these so that you're able to use them. So if you look up dplyr, you'll see that the first two hits on your web browser are going to be what looks to be a dplyr specific web page, and then this other entry that's going to be the CRAN repository for dplyr. So if you look at the CRAN repository's webpage, it's not very helpful and it's not very visually pleasing. It does have some vignettes as far as explanation of how to use the package, but if you open them, they're all text-based. There's not really much illustrations. So it's definitely not beginner-friendly when it all is said and done. So what I'll often do for CRAN packages is I will search first to see, do they have an actual website? And this kind of style of a website is what's known as a package down website. It's made by an R package that's known as package down, which basically in a couple clicks will generate you this nice, helpful website with the reference basically being all the different functions that are present within the package and how to use them, kind of that same information you get from the help page. But then it allows you to do little articles which are basically short walkthroughs of how to use particular aspects of that package. So this is kind of like my first stop when I'm trying to learn about a package and I have like no context of how to go about using it. Now, not all GitHub-based packages also have websites. It often depends on if their individual developers have implemented one or not, but the ones that you do find often tend to be these package down style websites as well. Now, you won't be able to find these usually directly by Google searching, but if you look at the actual GitHub repository page for the package, oftentimes there'll be a link to the documentation that's either present on the right sidebar, or in like the case of the autospectral package when I was looking at it last week, there would be like a situation where you'll see it linked in the actual README portion down on the bottom of the view. And then Bioconductor is kind of like halfway between a package style website and a CRAN kind of halfway between what we see for the CRAN repositories, all text-based and then a package down style website. Bioconductor does a really good job at documentation, especially with their vignettes. So if you find a package that's actually present on Bioconductor, when you're navigating to the web page, to scroll down, it'll generally be either a PDF or an HTML format of the vignette, and then just click on that, and it's kind of similar to the same thing you would see for a package down style website's article view. Additionally, for especially the Bioconductor packages, if you do go to the internal help files in R, and then you see the index page, you'll often see this user guide, package vignettes, and other documentations, and this will link you back to that same thing without having to search for it on the browser. All right, at this point, we have gone through most of what we needed to cover for today. All that really remains is we need to install the packages we'll be using throughout the course. I am going to be conscientious of time because we're at the hour, so this is going to be partly on y'all to make sure you get them installed. But I want you to emphasize, if you do encounter any errors at the installation process throughout, please take a screenshot, come back to the discussion page, post it, and we'll help you troubleshoot. So basically, I have this set up kind of like a challenge style of see how much you remember from the earlier portion of the class as far as like, how do I go about and troubleshoot? Is this package from CRAN? Is this package from Bioconductor? Is this package from GitHub? And then what installation commands would I use? Do I use the quotation marks? Similar things of that nature. So what I want you guys to do on your own time is basically say, here's like the first challenge. It asks, is the R package from PER? So what I would do if this was me trying to figure out how to install a package the first time is I will go ahead and copy the name of the package. I'll open up my web browser and I will Google. And you'll see Google knows I'm often looking for R packages, so it's popping up the initial suggestions. So I can see right off the bat, this kind of looks like a website. I'll go ahead and visit this one. And then right off the bat, it looks like a package down website. It mentions tidyverse, which makes me think it's likely CRAN. And sure enough, right there we have install.packages, and that's how we go about installing the per package. Now, I'm not a sadist. I'm not going to make you do this all on your own without help, especially if you're short on time. So what you can do is same thing. If you do to the next one, you have the little code drop-down option, and you can see that, yes, this package is located on CRAN. It gives you the website, and again, it gives you the install specific command for the CRAN repository, and it remembers the quotation marks for you. So there's a total of 20 different packages that will be used extensively throughout the course that I would like you to install when you have the time. So if you want to repeat this process for the Flowcore package, same thing I would do is I can search for it. I can search for Flowcore. I can see immediately there's a Bioconductor listing, which makes me think it's on Bioconductor. And sure enough, down here, you see the installation command that you would need to be able to install it from Bioconductor. And then we can go ahead and double check. And sure enough, it's from Bioconductor. So at this point, let's double click. There's about 20 different packages to install. I did update a couple things to make sure the install commands were correct this morning. There's a couple GitHub packages present that I'll let you figure out which they are. The main thing to remember is packages that you can find on Bioconductor and CRAN may also be present on GitHub. So oftentimes, if you are trying to identify something that's only available on GitHub, it's the absence of a CRAN and a Bioconductor website that kind of gives it away. And list of 20. So, congratulations, everyone. You have survived the first week of the Cytometry in R course. And my question for you at this point is, are you not entertained? Now, for those who were here in person over this last week, at this point looking around the room, this is where I see people are nervous. Everybody seems a little bit confused, and I just want you to know this is perfectly normal. None of this is intuitive. Why some things have parentheses, why some things don't, why there's 3 different sets of installation commands for 3 different repositories. None of this is particularly easy. We have all been in your shoes. It's something that we are going to learn just by having to be using packages over longer periods of time, troubleshooting, figuring these things out on our own. So what you're feeling is normal, and that you don't have to know how to do this by the end of today. It's something we'll be repeating throughout the course. So anytime we introduce a new package, or anytime we are actually using a package, I will give those same kind of reminders we did today as far as the installation commands, and the reason why we're using those installation commands, as well as the use of the library to attach things. As always, as you go through the installation, if you see anything that is not working, please take a screenshot, go to the discussion page. If you want to discuss things more in depth, there's also a section for that in the discussion page. The advantage of taking a class with so many flow cytometrists who are also trying to learn R at the same time is that if you have a question, odds are high there's at least 100 other people who have the same question, so you'll be in good company as we try to move forward. So this is a community effort. We're going to make sure that nobody gets left behind. Well, there may be differences in learning how to code as far as how quickly someone picks up. It's firmly on my belief that everybody can learn how to code just with a lot of practice as we go. And the wrap-up section is mostly a website-based one. It just kind of gives you the summary of what we've been looking at today. And the main things I would like you guys to be able to focus on is I want to preface what we'll be doing next week. We'll be working primarily with file paths, which is how your computer is able to tell R, go and look in this particular folder, find me these particular files, copy and paste them to a different location. So the selling point I have for it is recently I was trying to figure out how many FCS files I acquired during the course of my PhD. So if you're anything like me, you've copied and pasted it and duplicated different folders a couple times. So just looking up the total number of .fcs files on your computer is not really going to give you a good accurate measurement. But through the use of the file paths function, as well as the recursive argument, which we'll talk about later, I was able to get a list of all the FCS files that are present on my computer, get rid of the duplicates, and therefore come up with the number of FCS files I had processed during my PhD for my thesis defense at that stage. And let's go ahead and double click. And with that, that's all I have for you guys for today. If you have any questions, I'll be around for the chat for a little bit longer. There's also always for every week of the course additional resources. Now I know some of you prefer visual audio, while some of you prefer reading. So my additional resources section will always have a mix of both. So for additional resources I listed for this week, Tiny Tales was a good beginner-friendly blog spot talking about it. I also wanted to give a little more context about the Bioconductor project. So there was this good talk by Martin Morgan introducing the goals on YouTube, which I linked. And then some of you have some existing R experience and may be interested in some of the finer details. The R packages book by Hadley Wickham is very good. It may not be for somebody who's just getting started off, but if you have a little bit more experience under your belt, it's worth a read. And then the take-home problems are optional. You don't have to do them. Um, all this material I provided today has been pretty carefully curated. You may find an occasional bug here and there, but you're pretty much on a guided tour. Um, a lot of the learning process as far as building your own mindset of how to work with code, how to adapt and troubleshoot code, it takes a little bit of trial and error. So this is particularly good if you're trying to work with your own data sets. So in this case, I kind of just give you 3 problems to work around on your own time. You can discuss these on the discussion forum with others if you're running into trouble and need some help. But then you can also submit them to us via the pull request feature, and then I will provide you individualized feedback to help troubleshoot those problems as well. So we have worked with PicoQC to get it installed today, but we haven't really looked at what the functions are. So this kind of sends you down the road to look up some help documentation information about PicoQC and report back. The second challenge problem is basically I give you the list of bioconductor cytometry packages. I want some little bit of information about them, but I'm also really just trying to figure out what your particular interests are. We have a bioconductor focused lesson in the middle of the course, so I kind of want to know which packages we should focus on as far as adding and exploring in as far as what they do. And then the third one, which I didn't want to overwhelm everybody with today, is there's actually a fourth way of installing packages in R. It's the newer pack package. So once you have installed all the other 20 challenge ones, if you still want to install a couple additional packages we'll use later on in the course, I'm going to have you guys use the new pack package, try to install them, and then just tell me how different it looks. And with that, I would like to thank you for your attention. Everyone here at the University of Maryland is ecstatic about how many people are taking the course. A little bit nervous, but we're glad to be part of your learning journey. We are all in this together. We look forward to seeing you guys next week. And if you have any questions, I'm here for them. One question in the chat, any safety concerns about installing things from GitHub? There should always be a level of caution when you're installing something. So I will never just be like fully just install something, just install the entire GitHub contents to my computer at a given point in time. Generally, R packages are a little bit more in a self-contained environment. Most R packages are not downloading different scripts. So it's one of those situations that if you're encountering it, I would just double check. First off, is it somebody that is established? Is it something that's been present? And just kind of like judge it by hand at that point. So it's something that you can navigate accordingly. There's also an option of being able to download the package to your computer but not actually installing it. So then that way, within Positron, especially if you're opening it for the first time, it'll ask, do you trust the person who's providing this repository folder? And you have the option of selecting no, at which point you're able to kind of like be able to scroll through and figure it out. Obviously, if you're just beginning, figuring out if something's malicious malware is kind of hard to do, but I would say don't download. It's one of those things I haven't encountered. There hasn't been very many examples of actively malicious R packages on GitHub, just because we are a little bit lower on the totem pole as far as targets go, because people using R are generally working at targeted particular objects like analysis. There were not big computational developers or others, more attack surface at that point. All right, y'all. If there's no additional questions, I'll end this live stream here. If you do think of questions at any point this week, or if you run into any issues, the discussion page is there. I will be circling back sporadically over the next couple days. I have a bit of a paper to finish up, but I will get to things as I have time. So I appreciate your patience and your attendance, and I will see you all next week.